afternoon.
Sir,
Okay.
well, it is diseases. It is my honor to introduce the chairperson for the session, Professor R.K. Seth. Professor Seth has been the professor at the Department of Zoology for the longest time. I had the pleasure of attending his farewell just a couple of months ago. So Professor Seth's research interest lies in applied entomology and radiation biology. Professor Seth did his PhD from Department of Zoology, University of Delhi. He has been an advisor on a lot of important committees of Government of India, ranging from Ministry of Environment and Forest to some staff selection commission. He was also awarded a postdoctoral fellowship by the European Commission, which he pursued at the University of Bath in the UK. Welcome, sir. Our next chairperson for the session is Professor Sanjeev Kumar, who is currently at the Department of Biotechnology, Chaudhry Bansilal University. His research interests are in molecular and vector biology of malaria parasite. Professor Kumar did his PhD from Banaras Hindu University, and he is a recipient of several awards from NIH USA. So now I request the chairpersons to initiate the session. Thank you.
Should we start? Okay, good afternoon. We are going to start this session, and I will request to stick to the time to the extent possible, because we are slightly late. And our first speaker is the keynote lecture is by Dr. KL9 Bhatt. I am very pleased to introduce this gentleman, scientist F, and he is in INMAS. I have got a very wonderful association with INMAS. For the last 30-35 years I have been using gamma cell from INMAS. What's up? Okay. So, his area of interest is biochemistry, cell biology, radiation biology. Radiation is one common factor over there. People are using nuclear energy for various purposes, for medicine, for agriculture, for diagnostic purposes, etc. etc. And emerging medicine, development of prophylactic, mitigative, therapeutic drugs for nuclear and biological emergencies. And he has a wonderful academic record, research record. He has been visiting scientists in Switzerland. He has been visiting Fulbright scholar in USA. So with all this brief introduction, I would request him to present his talk.
Thank you very much to the chairpersons, Professor Seth, who have been our guide and mentor in radiation biology. As he said, we had long collaboration with the University Zoology Department, and I have privilege of teaching also as a guest faculty in Zoology Department. So, and I am thankful to the organizer also who gave me this opportunity to present my work here. And the work is 2DG as an anti-COVID therapeutic from an age-old molecule to a modern broad-spectrum antiviral therapy. So this is the work which we have done during the COVID. Being in a defense institute, we have been always, as radiation biology, as I said, and this molecule has come basically from there only as a adjuvant to anti-cancer, adjuvant to cancer radiotherapy. We repurpose this molecule as a anti-COVID drug. So, before going to that, I would just like to tell a little bit about the basic, how the biochemical pathways plays a role in cells, and then how they can be a very good target. You must be hearing a lot about that, what are the biochemical pathways and what can be, when they can be targeted and when they cannot be the target. When in differential systems, like in cancer, they are differentially expressed. In diseases also, they are differentially expressed and they can be the target. So I just want to connect this. This is from physics. You all have heard in basic physics the Kirchhoff's law. The current coming I1 plus I2 is equal to I3 plus I4. But long back, one of my friend shared this image, maybe somewhere in, you can assume like somewhere in Delhi, Old Delhi or anywhere in the city, he asked the question, can you apply Kirchhoff's law to this circuit? So it is very difficult circuit. I mean, it is very difficult to find out current is coming from how many sources, from where, and then current is being distributed to how many different circuits. So this was a very relevant question, but you must have known that this circuit is even more complex than that circuit, right? Whatever we have seen, I mean, a difficult circuit to find out that how much current is going in which direction. This circuit is even more complex, which is going on in cell every fraction of seconds in our every cell in the body. And the tribute goes to all those biochemists, all those scientists who really solved this circuit, and they know like when a molecule enters in the cell, where does it go, how does it benefit to life, how does it manifest in the form of life, and that's how we can I mean think that probably some of them can be the target and some of them can be utilized for that. So one of the old scientists, Otto Warburg, he, I mean, you all know like now the biochemical pathway for energy. Cancer cells basically rely more on glycolysis when they have to take the energy. I'll come, I mean, this was an understanding long back in 1930s when he did this work, and he found that cancer cells have compromised mitochondria. Then he saw that what basically, when the mitochondria is compromised and energy is basically, mitochondria is known as powerhouse of the cell. So these cells are taking energy from where? Then he realized that these cells have high glycolysis. And this phenomena is known as Warburg effect. In cancer biology and in biochemistry also, it is known as Warburg effect. Why Warburg effect? Like Pasteur effect, in the absence of oxygen when glycolysis goes up. But even in the presence of oxygen when glycolysis goes up, it's called Warburg effect. And this is a very well established phenomena. And what basically it does, so this was the background of that anti-COVID drug which I am talking about. That normal cells, when some cell gets damaged, to survive, they change their metabolic pathways, and that change in the metabolic pathway gives them impetus. I mean, they start proliferating very fast, and that's what happens in cancer cells. And when they are proliferating very fast, the glucose demand goes very high, and that's what happens. And 2-DG is one of the drugs which is anti-glucose. It is exactly like glucose, but at the position 2, the hydroxyl group is not there. So when you go in the glycolytic pathway, it gets converted into glucose 6-phosphate, 2-DG 6-phosphate, but that cannot be converted into fructose 6-phosphate. Why? Because at position 2, oxygen is required to convert a glucose into fructose, right? So because oxygen is not there, it cannot be converted into fructose 6-phosphate, and that's where it gets trapped inside the cell. It doesn't provide energy, and that leads to several other problems. So why glycolysis? Because single carbon metabolism, when that metabolism is very fast, what happens, the glucose input goes to glycolysis as well as P pathway, right? So P pathway is mainly anabolic pathway which gives you nucleotide metabolism, which gives you single carbon metabolism of fatty acid, as well as protein also it gives input. So when these things are required at very rapid pace, then the cell rely more on glycolysis. I will give you example. When we are in adrenaline gets secreted, when we are in a situation where we are in an emergency like fight, flight and 3F hormone, you know, we rely mostly on glycolysis. Why? Because glycolysis can give energy at very fast pace. It is not economic, but by using lot of molecule of glucose, you can produce energy at very fast pace, as well as these anabolic reactions also at very fast pace to come out of that emergency situation. So that is why glycolysis is important. This is the background. I mean, long back since 2 decades we have been working on this, and these two gentlemen who were our seniors, former director Professor Vinay Jain and Dr. B.S. Dwarkanath, he also had very long association with Delhi University. They worked on this molecule as a adjuvant to anti-cancer therapy, giving just before radiation, so that when radiation kills the cells, damage the cells, the energy will not be available to repair the cells. so the damage will be potentiated and the cancer cells will die more. This was the hypothesis, and it can arrest also the cancer cells growth. It will not kill the cancer cells, but it will arrest the growth of cancer cells, so the tumor will not grow further. This was the hypothesis, and they used it, and the clinical trial was also conducted at so many hospitals in glioblastoma patients, so it was already used in humans. And later in 2014, DCGI has given approval, recommended this drug for anti-cancer as an anti-cancer molecule. These are some of the clinical trial reports which they worked on and then published in a very high impact journals. The clinical trial reports, and this is some of the recent work where we use low dose of 2-DG to establish that if you give 2-DG initially while cancer is progressing, then cancer can be arrested at that initial situation, and this can be a very good drug for when carcinogenesis is induced. As I said in the beginning, that at the time of initiation of the carcinogenesis, metabolic switch happens. So if you target at that point of time only, probably we can stop the growth of the tumor there itself. But today we are going to talk about the antiviral, how it works as antiviral molecule. So like this was already established in the literature. It started coming some time back only. One clinical trial happened in 1983 in herpes virus, where 2-DG was used topically. I mean, because herpes happens on the skin, so it was used topically. Somehow it didn't work. That clinical trial was done in 36 patients, but it didn't work, so that work was stopped at that point. But recently, a lot of basic research was going on, and people have shown that when virus infects the host cell, because virus also needs very fast production of the virion molecules, a lot of virions. So that is also proliferation at very rapid speed, and that also needs similar glycolysis kind of switching from mitochondrial respiration. So that is why virus on infection, it hijacks the host cell metabolism, and there are a lot of reports. And recently in 2018, this paper got published in PNAs that rhinovirus, when it infects to the host cell, it basically switches the metabolism of the host cell, and probably that host cell metabolism can be targeted to as an antiviral therapy. So this is the problem in SARS-CoV-2. We all know that this is a very highly infectious virus, not like earlier any of the earlier virus. This was very highly infectious, and when Omicron came, that was 18 times more infectious than the first virus. So when we are talking, if I am infected, it will go and then you will respire it, you will inhale it, and then this will create the problem. So this is the entire etiology. I think most of us, this is one disease probably even a layman also got a lot of experience and knowledge about the disease, right? So these are the three different stages. Stage 1, when it reaches to the lung or even oropharyngeal region, and then it multiplies very fast there, and the virus multiplication happens at very fast rate. Stage 2, when it reaches to the lung cell, and you can see in the bottom how much damage to the cells it is causing. On the left side it is uninfected cells, and on the right side you can see the infected cells. So cells start dying, they will start, that is called classical in virology, it is called cytopathic effect. So cells will start dying because of the multiplications of the virus is happening at very rapid rate, you can see here. And this is what damage, and when the cells will die, what will happen? Inflammation will take place. So cytokine storm will come into this stage. And in the third, you can see here the stage 3, the damage has taken place and lung has completely gone. Because of inflammation, a lot of fluid comes in and then accumulates, and that is the situation when recovery of the patients become very difficult. People are shifting on the ventilator and they are dying. So these are the 3 different stages. How 2-DG can work in which direction? You can see here that the virus multiplication is coming out from the cells is very fast, all these viral molecules. And what basically this has? It has only 3 molecules, lipid, protein and carbohydrate little bit, because the modification of protein is happening, and then nucleic acid. So these proteins are phosphoproteins, so they need ATP, glycolysis is required there. Nucleotide synthesis, I told, like that is also need glucose there, and then even lipid also need a single carbon synthesis. So that is why for fast viral replication it needs a lot of glucose here in this molecule. So what basically happens inside the cell, you can see here this is a normal cell. It is most of the glucose when taken in, it goes to the TCA cycle and then normal process goes on. However, in the infected cells, what happens? That once it infects, it damages the mitochondria. Viral infection damages the mitochondria. It actually basically nitrosylates the mitochondrial protein. So majority of the mitochondrial protein, they will not be able to perform normal respiratory functions. So not completely damaged, but partially damaged the mitochondria. And that allows more viral infection. Actually, it is a vicious cycle. So what happens that during that situation, if cell has to survive, or the virus reprograms the host cell metabolism by integrating its genes, and that's what it does. Lot of glucose transporters will increase on the surface. So it will invite more and more glucose to get accumulated inside the cell, which will be utilized for more and more viral replication. So this is basically what happens in case of viral infection. So on stage 1, you can, by targeting 2Dg, you can arrest there the viral replication. In stage 2 also, because you know inflammation, and from anti-inflammatory to pro-inflammatory, the inflammatory cells also become more glycolytic. See, if you inhibit the glycolysis, macrophage can't do phagocytosis. So glycolysis is intricately required for even inflammatory functions also. So if you inhibit glycolysis in inflammatory stage, it will inhibit the inflammation as well. This is a very well established phenomenon in the literature. So you can go and check that. So so this at stage 1 and stage 2 both you can arrest the COVID. So this is the molecule you can see here. I mean, this is simple glucose molecule. At position 2 both are H and H. So this becomes very close to mannose also, you know, and in mannose and glucose they are epimer, right? At position 2 OH is below, and in mannose OH is up and H is down. So this is how this acts as a, this mimics even mannose also. We will tell why mannose is important in later part of the thing. So it is exactly at C2 position only the oxygen is gone, so it behaves exactly like glucose outside the cell. I mean, body cannot recognize, differentiate between glucose and 2DG outside the cell. But when it goes inside the cell, as I said, it will get arrested there, and that's how the problem starts. I mean, it will arrest more in cancer cells or virally infected cells. We will show here. So as we discussed already in detail that upon viral infection, it hijacks the host cell metabolism, and then the cells become more greedy for glucose. Lot of glucose is required, and you know, like in infection also this happens. Infection leads to insulin resistance, and more glucose comes in the bloodstream, and that's why more glucose comes in the bloodstream, because it has to facilitate the macrophages and inflammation to sort out, to come over that infection. And this is a natural phenomenon, happens in all of our body. So this is the thing, like glucose, more glucose requirement is there at that point of time. Then as I said, like ATP, rate of production of ATP is much faster in glycolysis. Like if a single molecule of glucose, let's say, take 1 minute from going from top to bottom, I mean conversion to carbon dioxide and water through oxidative respiration, it will produce, let's say, 36 molecules of ATP, whatever as per the classical biochemistry. But if in the same 1 minute, even if it is not going to the glycolysis, into the mitochondrial respiration, if glycolysis is high, 50 molecules of glucose can be processed. So what will be the total net gain of ATP? That will be 100 molecules, right? So as compared to 36, you are able to get 100 molecules of ATP. So that is why in emergency situations, glycolysis is basically required. When we are running, glycolysis is basically required, because oxygen is also not available at that point of time. Then other than that, I mean, SARS-CoV-2 infected cells, metabolic plasticity is very important in normal cells. It can switch over between the glycolysis and mitochondrial respiration. But in case of virally infected cells, as I said, virally infected cells and cancer cells, mitochondria is damaged. So glycolysis is the only pathway which is providing energy in that to do any function, either to synthesize the virus or to survive the cell. So targeting becomes easy. And these are the very initial evidences. We also started our work in March 2020 when COVID came in India. And you know, this paper got published in May 2020, suggesting that they have done simple proteomics of, they infected the host cell, and they say that what are the pathways which is getting activated, and all these pathways can be targeted. So this is a very basic work in which they found glycolysis is also upregulating, so glycolysis can also be targeted, and they showed also 2DG, using 2DG the viral replication is going down. So here, and this is another work which will suggest, I mean in common public we know diabetes were more susceptible. Why diabetics were more susceptible? Because they have high level of glucose continuously circulating in their body. And this paper published in Cell came little later in 2020 end, which suggesting that if glucose level will be high, it will facilitate the viral infection in the cells, and that will lead to more and more, I mean SARS-CoV-2, the body will facilitate the SARS-CoV-2 more infection. So this these two work got published, and parallelly we were also doing this we already discussed about. And then coming to then what we have done, how we found out that probably 2DG can be a good molecule. So these are the results. You can see here that this is uninfected cells and this is infected cells. This green color is a fluorescent 2DG, you can say, because it's a fluorescent molecule. And you can see here in uninfected cells the accumulation of 2DG is not very high, but in infected cells accumulation of 2DG is very high. This is the quantitative data, and you can see here the glucose uptake and lactate production and ATP production also goes very high when the infection happens, right? So 2DG becomes and upon infection you can see all the GLUT1 goes very high, which can trap the glucose at very low level of glucose. These are very efficient glucose transporters. At low level of glucose they can transport, and GLUT3 as well. So the level of GLUT1 and GLUT3 goes high, GLUT4 also goes high. So they all are pushing more and more glucose in the virally infected cells. And then the enzymes also which are processing glucose, that has also gone up. So you can see here that when we infected SARS-CoV-2 in Vero E6 cells, we found that all the glycolytic markers are going high. And this is a very important thing. So people say like this is a normal glucose, it will go to every cell. But you can see here this experiment of us, we co-cultured two cells. The one cell which takes very low, very minimal viral infection, that is Vero cells, they are isogenic cells, and the Vero E6 which is modified and it takes high viral load viral infection. So these Vero cells are labeled as red, you can see here, and Vero E6 cells are not labeled. So we have given, they are co-cultured, and when we have given glucose 2DG, you can see that all those cells which are not looking red, they have accumulated more and more 2DG, right? So this suggests that even in lung cells, when some cells will be infected and some cells will not be infected, it will be mainly infected cells which will accumulate very high amount of dose. And in drug, when we are designing drug, this is a very important thing. The drug is targeted, I mean, this is using the inherent phenomena of the body or the body mechanism to target the drug. We are not doing any other targeting. We are not designing the molecule in such a way that it should go to only the targeted cells. It is using the inherent phenomena to reach to the target cells only, and that is what basically happened here in co-culture. So in a simple model we have tried to explain here. You can see here that these are only virally infected cells and in lung cells, and it is getting accumulated more as compared to uninfected, where the level of 2DG is very less. And you can see here that as compared to uninfected, this is a quantitative data that the 2DG is very high. Then the other things come, because you know in cancer biology people have been thinking that 2DG is to kill the cancer cells. So they said that probably 2DG will kill the other cells also, which is not required probably in biology. But it is very well known that it will arrest the growth of the cells. And our normal cells are not proliferating, right? Our blood cells are only proliferating. Some of the cells are only continuously proliferating. Rest of the cells are senescent, I mean they are not proliferating. So it inhibits the growth of those cells, which we have shown through here, that it is not basically killing. Clonogenicity is also not changed, and it is basically not killing the cells. What it is doing, it is just stopping the proliferation of those cells. Then we checked its inhibition. This work was done during lockdown. It was done in May when I went to CCMB and conducted these experiments. because that time virus was only available in CCMB and NIV. These were the two institutes. So we conducted these experiments at CCMB in B.6 variant, the very first variant which was detected in India, and we found that the effect is very good. And later we did in B.1.1 variant, which we established in our BSL-3 lab. First we established the BSL-3 lab at INMAS, and then we established these cultures also. We found that this is equally effective here also. In Delta and Omicron variant also, it is equally effective. And you can see here the production of the proteins also. I mean, the N protein, nucleic acid protein and spike protein also goes down when the 2DG is given to the cells, infected cells. Then in fact you can see here very well, you can see here this is an infected lot of cell that you can see, which is called CPE. Classically in virology it's called CPE, and at 1 millimolar and 5 millimolar you can see here the effect is completely gone. How much time? I'm trying to complete quickly. So why I said manos, you can see here with increasing glucose, like in case of diabetes, people thought that probably 2DG will interfere with glucose and effect will go down, but you can see here that at very high concentration of glucose also the effect is not compromised. 2DG effect is not compromised, but mannose is able to compromise its effect. Why? Because mannose at very low concentration it is taken up. So 2DG at very low concentration it will be taken up and it will be able to affect. So one hypothesis was directly by inhibiting glycolysis, by anabolism, it is able to sequester the virus. But you can see here what we found in the next, when we have taken out the virions from 2DG treated cells and untreated cells, and we calibrated, we equalized the concentration and then infected without 2DG, you can see the virions are not able to infect the new cells. Why this is happening? What is the reason? You can see here that 2DG it mimics mannose, and mannose is required for what? Basically for glycosylation of the protein, and all the proteins on the virus surface, they are glycosylated. So and glycosylation is like address on the letter. So if you write a wrong address, it will either not reach to the right place, the letter will not reach to the right place, or it will be again go back to the post office. So this is what basically happens. You can see here the two glycosylated moieties become deglycosylated, and that is why probably virus is not able to bind. So this is, we proposed a model, and you can see here the virions are not able to, the virions produced from 2DG infected cells, whatever, like let's say 90-95% inhibition is there. So whatever 5% virions are being produced after even 2DG treatment, they are not able to infect the neighboring cells. And that is what we proposed in this. This is a common normal thing which is happening. But in 2DG treated cells, first thing it will reduce the viral production, and whatever virus will be produced, they will be with deglycosylated protein, so they will not be able to infect the neighboring cells. So overall it is able to infect, stop none of the other drugs. They have two mechanisms of working. They don't have even direct targeting of the cells, but this drug does have. This study got published in Life Sciences, and you know, as I said, like this has a very good anti-inflammatory effect also. So it will be able to capture at two stages, stage 2 of the COVID also. And this is the PNA study which got published, where they showed that 2DG works very well in rhinovirus also. So this we already explained. Based on this, we did clinical trial. We went to phase 2 and we did clinical trial. And all the patients who were recruited in this clinical trial, they were actually at stage 5. WHO has given an ordinal scale. So 5 means all the patients who need oxygen. They are not only hospitalized to the hospital, but they need oxygen supplementation also. That means oxygen is below 94% saturation. So they need oxygen. So all such patients were recruited, and when we given them different doses of the 2DG, we found that 90 mg per kg per day is very effective dose. And you can see here, this is the parameter. This like time to achieve SpO2 94% was from 5 days, it has came down to 2.5 days. And then time to discharge also reduced, time to clinical recovery also reduced, and then time to vital signs normalization, time to achieve 1 point on WHO ordinal scale, and time to achieve at 2 point at WHO ordinal scale. Both has gone significantly down. Based on this, actually we have gone to phase 3 also, and we performed this study in 220 patients. These are some of the key things, and you can see by day 3, significant number of people is coming out of showing 2-point improvement, and this is 1-point improvement, which suggests the requirement of oxygen, the damage to the lung cells, which promotes the oxygen requirement at that point of time is basically that is working very well. Anti-inflammation is working, it is targeting inflammation very well, and the drug is able to give relief to the patients very nicely. And this is the ICU patients, you can see as compared to these red lines. I mean, we compared everything. This was used as an adjunct. So in control, all the patients were given remdesivir, favipiravir, all the drugs. In 2DG treatment, they have given along with these antivirals, 2DG has been given, and you can see as compared to them, the patients coming out of ICU is very fast as compared to standard of care where all the antivirals have been given. So this study also got published recently in BMC Infectious Diseases, and 21 this got the emergency use approval from DCGI, this drug, and it is available in the market now, and it is very simple. I mean, it comes as a glucose powder, so you can sachet you can dissolve in 100 ml of water, and the people can drink it. And these are the people who contributed immensely. Like when we do research in the lab, it is just like we only have to manage budget and everything. But when you are going to industry, we had to rope in one pharmaceutical industry, Dr. Reddy's lab. So Dr. Reddy, actually our chairman and secretary DRDO that time, he is the person who converted this possibility into reality. He is the man behind this who helped us a lot, and meeting all the challenges, funding and everything. So thanks to him, and then all the scientific team and my students. During the lockdown, labs were not open, but we had got requests and then permission. So all this work was going on during the full lockdown also in my lab. So thanks to all the students and my collaborators who worked well, and this is the team from Dr. Reddy's lab who also worked day and night during that time to bring this molecule to the public. And finally this got approval in May 21 to use in emergency situation in hospitalized patients. And I am happy to share, actually I am carrying the pouch also. It is very privileged for any scientist, like if your research from the lab, it comes to the public to use. So this is thank you very much.
One or two questions.
What is the effect of 2G and RBCs?
RBCs. See, we have not checked, but as you said very rightly, we have been thinking. I think you are asking from glycosylation, deglycosylation.
Yes, because RBC don't have mitochondria and nucleus. They directly depend on
glycolysis, right?
And also they carry oxygen.
Right. So as far as this oxygen carrying is concerned, that was one of our main parameters. So that got even better. But our main concern is what happens if glycolysis is able to modify the glycosylation. I mean, RBC only carrying this ABO blood groups, right? So our main concern is from side effect perspective, from that perspective, and really we have not checked. As far as the number of RBCs and number of WBCs are concerned, they are absolutely normal in the control patients as well as treated patients. So this is the clinical parameter I am talking about. We have now more than 400-500 patients treated with 2DG, and they are absolutely fine. I mean, there is no problem as far as the numbers are concerned. But yes, that concern is already there. We are working in that direction to check whether there is some glycosylation changes are happening or not because of continuous use of 2DG for a long time, like 15 days or something.
And also, like, calorie restriction is good idea.
Right, for cancer it is very nicely working. Right. So what work I have shown you published in European Journal of Cancer and PLOS ONE, there we are using it as a dietary supplement. 0.2% continuously in water. The mice, they drink that water continuously whole day, and tumor formation is much less. I mean, first lot of mice, they don't develop tumor on implantation. Those who develop, they develop very small tumors. So that is a very, I mean, that work is already published in class 1 and urinary general cancer. You can check, search on the Google, and you can find it. That is there. So it is working very nicely as a dietary supplement.
Okay, thank you.
I think there was another guy. You wanted to ask something?
Yeah, please.
Please.
Can this 2DG differentiate between the infected cells and non-infected cells?
Yeah, that's what I showed there, one of our experiment, where, as I said, like the transporters on the surface will be very high. So in normal cells, only very few transporters will be there, so it will go there also. But as compared to uninfected cells, when in infected cells, they have very high number of transporters on the surface. So they will accumulate much high. The other thing is, like when we give 2DG, whatever dose we are giving, it reaches only 0.5 millimolar to the blood, right? But glucose is already 5.5 millimolar in the blood. At 100 mg per deciliter, it is 5.5 millimolar in the blood. So it will not, in normal cells, it will not have any problem. But in infected cells, the transporters which are expressed on the surface, they have very high affinity with the glucose. So they will sequester more and more 2DG. So it will go mainly to uninfected cells. Infected cells.
But there is no competitive inhibition.
Yeah, that's what I showed. Yeah, I showed a data. I showed a data. I mean, we have gone up to.
It has almost like equimolar, right?
Right.
One is 5, 5 and one is 5.5.
Right.
So then
No, it is 0.5. 2DG at the dose which we are using, it reaches 0.5. Almost 9 to 10 fold difference. 10 fold lesser than this.
Yeah.
So when we showed at 10 millimolar glucose, then also it is able to inhibit that viral replication. So in media we used 10 millimolar glucose and we used the same 15 millimolar glucose and used the same 5 millimolar glucose. In vitro I am talking about. It is able to inhibit the viral replication there also.
Dr. Anand, I have this side.
Yes, sir.
So 2DG is a natural molecule or is a man-made molecule?
Sir, it is synthetic, man.
Synthetic. So not found in nature.
Sir, it is found in nature, but that position 2 is methylated. It is believed that some of the fruits, they do have this molecule, but at position 2 there is methylation. So there also it works like the same as it is working here, but this is a synthetic molecule.
Can you name those fruits? So
actually, I am also searching for that, but I have heard that there are some herbs which people in Brazil and South America, they use it as a tea. So I am also searching. I just have heard that they have similar kind of dietary effects, but I don't know exactly.
Thank you.
Like methylated mannose.
Right. Yeah, I mean anything which gets methylated at position 2 may work like that.
So in fact, Dr. Anand, you are the real hero behind the real situation, because we all remember the COVID situation and 2DG all of a sudden came in picture. And thanks to God and thanks to you people.
Thank you.
First heard this name 2DG from Professor Vinay Jain.
Yes, sir. He is the father of this study.
Now, to kindly felicitate Dr. Anand Narayan Bhatt for delivering such an exciting lecture.
Can I also have this pleasure to come with?
Oh, please.
You are such a real hero. I think we need a picture. I will.
Wait.